Welcome, everybody. My name is Theresa, and first I would like to thank the organizing committee for giving me this opportunity to speak today. I'm going to present some of our recent results about the association of vascular burden and Alzheimer's disease pathology with changes in functional connectivity in persons at risk of Alzheimer's disease. It has been proposed that Alzheimer's disease is a multifactorial disease that is not only characterized by Alzheimer's disease pathologies such as amyloid beta and tau, but also by early vascular dysfunctions, which may lead to later brain changes such as alterations in functional connectivity. However, this hypothesis was built so far mainly on cross-sectional studies, suggesting that cardiovascular risk factors in Alzheimer's disease biomarkers are associated with abnormal brain resting state functional connectivity in aging and Alzheimer's disease. In the current study, we aim to investigate whether vascular risk factors as well as cerebral A beta and tau deposition are associated with changes in functional connectivity over time in individuals that are still asymptomatic but at higher risk for Alzheimer's disease. In total, we include 247 cognitively unimpaired older individuals of the Prevent AD cohort. All individuals had a family history of Alzheimer's disease, which set them to a 2 to 3-fold higher risk of developing Alzheimer's disease later on in their lives. The participants had a mean age of 63 years and 75% of the cohort were female. At baseline, we assessed from all individuals the concentration of the plasma lipids, total cholesterol, as well as of its subfractions of the so-called good HDL and bad LDL cholesterol, as well as the systolic and diastolic blood pressure. During the course of the longitudinal study, with a follow-up of up to 4 years, a subset of 91 individuals underwent also a PET scan, where we assessed beta amyloid as well as the tau deposition in the brain. With regard to resting state fMRI, functional MRI, which was performed annually, participants needed to have at least 2 valid resting state fMRI scans, so which brought us to 240 scans at baseline, 232 scans at follow-up year 1, 185 scans at year 2, 130 at year 3, and 78 scans at follow-up year 4. So that we had in total a number of 865 scans with an average follow-up time of 3.5 years. Those scans were segmented using the Schaefer-Yeho parcellation atlas. Then the functional time series were extracted from each of those parcels, and individual matrices with positive correlations between the time series of each parcel were created and Fisher set transformed. Finally, the average correlations between the parcels of predefined functional networks that are here displayed in the multicolored squares, as well as the whole brain displayed here in the big red square, were extracted, representing indirect measures of network-based and global resting state functional connectivity. This procedure was also repeated with 2 other previously validated functional parcellation atlases. The one from Wurst et al., it's called the MIST parcellation atlas, and from Power et al., the Power atlas. Statistically, we were finally running linear mixed effect models, including all vascular risk factors, as well as age, sex, vascular medication, and mean frame displacement as covariates. And as random effects, we included individual intercepts and slopes. However, we had a high collinearity between total cholesterol and LDL cholesterol, and therefore we divided those 2 lipid measures into 2 models, in model A and model B, together with the other vascular risk factors. Turning to the results, we show here the t-value results of the linear mixed effect models. We found that across all functional networks, higher total and LDL cholesterol levels were associated with a decrease in functional connectivity, specifically within the default mode network. And with regard to blood pressure, we found that higher diastolic blood pressure, but not systolic blood pressure, were associated also with a decrease in functional connectivity within several networks and across the global brain. In contrast, we could not find any associations of beta amyloid and tau deposition with a longitudinal change in functional connectivity, neither in one network nor globally. So the findings about the associations between the first two that I mentioned, between the cholesterol levels and the default mode functional connectivity changes, as well as between the diastolic blood pressure and the global functional connectivity changes, were also replicated when we used the other two parcellation atlases, the MIST and the Power Parcellation Atlas. And those same results we show here again, but in a longitudinal plot where we divided the continuous total and LDL cholesterol levels, only for a better visualization, into tertiles of low, medium, and high levels, which we color-coded in green, orange, and red respectively. It is interesting to see that individuals with a lower cholesterol level tend to show an increase in default mode functional connectivity, while individuals with a medium cholesterol level show a plateau, and only individuals with high levels of cholesterol tend to decline over time. So across the entire sample, regardless of the cholesterol levels, we didn't actually see a change in the default mode network functional connectivity over time. However, in those individuals that have high vascular or high cholesterol burden. And when we look at the association between the diastolic blood pressure and the changes in global functional connectivity, we see that individuals with low and medium diastolic blood pressure levels show rather an increase in global functional connectivity, while higher diastolic blood pressure levels are linked to a decline of those slopes down to a plateau. So here overall across the entire sample, we do see a significant increase in global functional connectivity over time, but as I said, not in the presence of higher levels of diastolic blood pressure. Interestingly, it has been recently shown that in the course of aging, both the default mode network and the global functional connectivity seem to increase before an age of 67, which is followed by a plateau phase between 67 and 74 years, and a decline in functional connectivity after age of 74 years. So regarding the age distribution of our cohort, which I show here, we would then rather expect an increase in functional connectivity over time. However, we do see this kind of trajectory only in the participants that have a low vascular burden. And in contrast, in individuals with medium or high vascular burden, it shows a plateau or even a decline in functional connectivity. Thus, one can hypothesize that the presence of higher vascular burden preludes the tipping point where neurons, maybe specifically in hub regions, get overwhelmed and which result finally in a functional disconnection. And in other words, also the individuals with higher vascular burden had on average the same age as the individuals with lower vascular burden. They show kind of an accelerated alteration process in functional connectivity. But this hypothesis should be definitely further investigated. Moreover, the fact that we didn't find any association between Alzheimer's disease pathology markers and changes in functional connectivity may reflect that vascular burden precedes the effect of Alzheimer's disease pathology on resting state functional connectivity. And therefore, we aim to continue to follow these individuals to assess if and when Alzheimer's disease pathology starts to impair the functional connectivity, and if vascular risk factors and amyloid and tau burden have an additive or maybe a synergistic effect on brain connectivity. And with this, I would like to stop and importantly mention that most of the Prevent AD data will be publicly available within the upcoming months. So please go and check it out. And with this, I would like to thank you very much for listening. And a special thanks goes to my supervisor, Dr. Sylvain Villeneuve, and the whole research team. Thank you.
In this talk, I'll be focusing on transcriptomic analysis of Alzheimer's disease associated with brain metabolism, hypermetabolism. Alzheimer's disease starts decades before clinical symptoms appear. One of the most prevalent neurodegenerative diseases is thought to affect approximately 5% of those aged 60 years and above worldwide. It is the most common form of dementia. Clinically, it is characterized by a severe decline in cognitive functioning and defined neuropathologically as seen in this figure, a progression of amyloid plaque, neurofibrillary tangles, and loss of neurons. Loss of low glucose utilization regions of the cerebral cortex marks AD and is informative for clinical diagnosis. This is measured by FDG PET. Patterns of hypermetabolism can be seen in at-risk individuals decades before the development of symptoms. Despite the clear link between metabolic changes measured by FDG PET and risk for AD, it remains unclear which mechanisms are responsible for driving these changes. Our aim is to molecularly characterize the patterns of regional hypermetabolism found in patients with AD and to identify molecules that protect certain brain regions or make others more vulnerable from spatial transcriptomic patterns in normal brain. Here is a general study overview. To bridge between regions and genes, here we integrate FDG PET hypermetabolism map with the Allen Human Brain Atlas microarray gene expression dataset. We identify genes with spatial patterns similar to that of lower glucose metabolism in the human brain. For further data analysis, we also look at gene ontology enrichment and cell type marker enrichment. So the first step is we performed a voxel-wise meta-analysis of the FDG PET studies that compared at-rest AD with healthy controls. A literature search was conducted on studies from January 1985 to January 2012. In this meta-analysis, we had 27 FDG PET studies, and it consisted of 915 AD patients, 715 healthy controls. Activation likelihood estimation, or ALE maps were created using Brain Map Ginger ALE application. The software assigns each voxel an activation likelihood estimate, which is equal to the probability that at least one of the reported peaks of hypermetabolism is located in that voxel. So a voxel-wise threshold of 0.006 was used to determine if a given voxel was inside an AD-associated region of hypermetabolism. So for the microarray gene expression dataset that was downloaded from the Allen Human Brain Atlas data portal, this involved 6 donors, which were 5 males, 1 female, with age ranging from 24 to 57 years. We used probes that are correlated with RNA-seq, and expression values were mean averaged for genes with multiple probes. So therefore, in total, we had 31,452 probes, which was averaged to 15,143 genes. And we restricted the sample to the cerebral cortex. For each donor and gene, expression values were compared between samples inside and outside of the hypermetabolic regions using the man-Whitney U test. And from the Allen Institute, they provided the MNI coordinates, which were used to help expression values into the voxel space of the FDG PET meta-analysis. Next, we performed gene ontology enrichment of the genes in the hypermetabolic regions. Genes were sorted from the most overexpressed to underexpressed in regions of hypermetabolism. So in this ranked gene list, the area under the curve, or AUC, was used to test for gene ontology terms that were significantly enriched in either directions. So in terms of the results, looking at the meta-analysis of the FDG PET studies in AD, we identified the following regions showing lower glucose metabolism in patients versus controls. And these regions included posterior cingulate gyrus, middle frontal region, angular gyrus, and middle and inferior temporal regions. So in this figure, we see the cortical surface views of the ALE meta-analysis results, which is lower glucose utilization of AD versus controls. And this ranged from low, which is seen in the shades of yellow, to high, which is seen in black. Next, across all 6 donors combined, we looked at the 15,143 genes that were tested, and we found that 99 genes were overexpressed and 51 genes were underexpressed. There were variabilities across each of the 6 brains. So therefore, we tested each brain separately. One donor influenced the atlas-wide signal for spatial expression overlap with the FDG PET-derived map seen across all 6 donors when they were combined. So this one donor was the outlier, which was driving this signal, which was, he was a 39-year-old male. So in terms of this donor, overall, there was 747 genes who were differentially expressed, which was not seen in the other 5 donors. So looking a bit at the raw data at the gene level, the most overexpressed gene was the RPL35, showing high expression in the hypermetabolic regions compared to non-AD associated regions in this particular donor, which is highlighted in the red box. The y-axis is the expression levels, and we can see that the RPL34 gene is highly expressed in the hypermetabolic regions, which is not seen in the 5 donors. Next, we performed Go enrichment analysis of the transcriptomic-wide results from the outlier donor. And the strongest Go enrichment signal is SRP-dependent co-transitional protein targeting to the membrane. So this Go term also contains the overexpressed gene, which was found in the previous slide, the RPL35. These genes encode proteins of the systolic ribosomes and are involved in targeting protein translation to the endoplasmic reticulum. In this figure, on the y-axis is the ranked expression of SRP genes. So overexpression is near the top of the y-axis and underexpression is near the bottom. And the x-axis is each of the donors. So we can see that in the highlighted red box of the outlier donor that the majority of the SRP genes are clustered towards a high-ranked differential expression compared to the other donors. Using the transcriptomic data in the outlier donor, we estimated lower portions of neurons and more microglia in the hypermetabolic regions when compared to the other 5 brains. So in the 5 brains, we saw that the microglia marker genes were expressed at low levels in the hypermetabolic regions. However, in contrast, the outlier donor had microglia marker genes were expressed at higher levels. And this can be seen in this figure, which is similar to the previous figure in the last slide. The y-axis is the ranked expression of microglia marker genes this time, and the overexpression is on the top of the y-axis, and underexpression is near the bottom of the y-axis. And the x-axis shows the donors. So we can see in the highlighted box, the outlier shows that the majority of the microglia marker genes are biased towards the higher ranked differential expression compared to the other donors. So as a follow-up analysis on the microglia differential expression, we used previous supplemental data of microglia cell expression in both human and mouse tissue. So this supplemental data was already published, and we did bioinformatic analysis on this data, which demonstrated that the SRP genes increase in expression across AD-associated states of microglia activation. So in the top figure, which is the human data set, 55% of the SRP genes progressively increase across AD-associated states from no pathology to late pathology. And in the bottom figure, which is the mouse data set, around 75% of the SRP genes progressively increased from normal cell to AD-associated microglia cell clusters, known as in this figure, group 3-DAM. So the expression for each gene is Z-scored with high expression in red and low expression in blue. And we can see that the high expression of these SRP genes are found in the later stages of AD. So in conclusion, the FDG PET studies of AD show consistent patterns of hypermetabolism, which has been seen in literature. Of the 6 normal brains tested, only 1 demonstrated a strong spatial association between gene expression and AD-associated hypermetabolic map. So this was driven by genes located in the GO term, which is the SRP-dependent co-transitional protein targeting to the ER. So we estimate that higher microglia and lower neuron proportions in hypermetabolic regions in this particular donor. So it is possible that this donor may be in the hypothetical stage of preclinical AD, but more research needs to be done to confirm this. So most of the SRP genes that have stage expression patterns, they increase across cellular and pathological AD-associated states in microglia. So by taking a molecular perspective of the FDG PET patterns, we highlight a unique donor with microglia dynamics. So I'd like to thank Derek, Deborah, Elena, and Julie for their work on the gene expression in FDG PET meta-analysis, and also like to thank the organization of the Human Brain Mapping for this opportunity to present our work.
Hi, my name is Pablo Damasceno. I'm a machine learning specialist at the Center for Intelligent Imaging at the University of California, San Francisco. And today we'll be talking about how the connectome can help enhance the predictions of future tau accumulation in Alzheimer's disease. I'd like to start off by acknowledging our collaborators, Professor Ashish Raj, Professor Gil Rabinovici at UCSF and his postdoc Renaud Lajoie, and the members of Eli Lilly who provided us with the data. So the motivation for this study is a recent discovery that the pattern of tau accumulation is extremely related to the pattern of atrophy that follows through time, and that this pattern of atrophy is what is causing the cognitive decline that's observed in Alzheimer's disease patients. So given these results, it's not surprising that drug companies are focusing on clearing the amount of tau, the tau accumulation in the brain as a way of creating a drug. So predicting the amount of tau that accumulates in the brain allows us to create a synthetic control group against which the measurements of tau in the therapy group would be compared. So the data we received from Eli Lilly was a group of 60 MCI subjects and 28 Alzheimer's disease patients. The data was composed of amyloid atrophy and tau PET, and both baseline and 18 months follow-up were present. This was already registered and segmented in the Desikan-Kiliani Atlas. The question they were interested in was, can the amount of tau in this period of 18 months be predicted with very high accuracy? I'll jump straight to the conclusions here that not all brain regions are created equal, and that some regions are much more predictable than others. Identifying those reliable regions can lead to a much stronger prediction. So the model that we used was the network diffusion model created by Ashish Raj and collaborators, where the idea here is that the amount of tau accumulating in one brain region is basically dictated by the amount of tau that was there to begin with at baseline. And this is also modulated by a Laplacian, which comes from the structural connectivity of healthy brains measured by tractography. And what this is telling us is that if a given region initially has some amount of tau, this region will influence the neighboring regions according to the structural connectivity of the brain, and the amount of tau will basically propagate following the Brecht staging observed empirically from data. So what we did was we basically fit this model for the data and tried to predict the amount of delta tau in each brain region. What we observed was that if you try to predict the delta tau in every single brain region per subject, we don't have a very strong correlation between our predictions, here shown in the y-axis, and the data coming from Eli Lilly, here shown in the x-axis. Our R-squared was of 0.25, which is not very remarkable. Now, if instead of focusing on every single brain region, we focus on a collection of regions, and in here what I'm showing in the next slide will be a result for all cortical regions average, what you see is that the amount of our prediction becomes with an R-squared of 0.55, which is a lot better, showing that if you try to make a prediction for the combination of regions composing the cortical subset, our prediction has a lot stronger power. We can still do a lot or somewhat better by, instead of looking at all 68 regions, we use our prior knowledge about the way that Alzheimer's, the tau accumulation in Alzheimer's disease followed the Brecht staging. And what we found was that if we look at the combination of regions composing the Brecht5 subset of regions in the brain, we have an even better prediction. So our suggestion here was that if instead of looking at our regions, you start looking at important subsets, your predictive power seems to be getting stronger. So can we do even better than this? And what I will show next is that we can if we find a way to select even further a subset of brain regions out of those 34 that make up the Brecht5. What we did, or what we used in order to get there was, again, the network diffusion model. And what we did was to start by seeding the brain uniformly in all regions up to Brecht stage 4. We then used the network diffusion model to evolve these regions in time and calculate how much tau was accumulated in all the brain regions. Then we took all these brain regions and we ranked them by how much tau increased over this time that we have just measured. And what we found was that there was an interesting kink in this histogram showing that there is a natural way of selecting a group of regions where you get the best bang for the buck. Finally, we use this subset of regions to make a prediction just like before, where you're trying to predict the mean amount of tau accumulated in these regions. What we found was that if you take the top 8 regions predicted by the network diffusion model to have the highest accumulation of tau over time, this gives us a very good prediction for the Alzheimer's disease cohort, with a still very strong prediction for the MCI subjects here that are being shown as circles. This suggests that even though these regions give a tremendous amount of predictive power, the same regions might not be as useful for MCI as they are useful for Alzheimer's. So in order to test this hypothesis, what we did was to make a subset even further, going even further, we made a subset of the first 4 regions from the subset of 8. And what we observed was that this subset of the top 4 regions make a remarkable prediction for the amount of tau accumulating in the brain with an R-squared of 0.71, whereas also remarkably, the R-squared for the MCI cohort decreases by an extreme amount. This suggests that while those 4 regions are particularly useful for predicting delta tau in Alzheimer's subjects, this group or this subset of regions is not as important for the cohort of MCI subjects. If we then just make a shift of, instead of taking the top 4 regions, we look at the next 4 regions, which are not as advanced in terms of the network diffusion or the tau that would be diffusing and accumulating in the brain, we see basically the opposite happening, where now the MCI cohort has the strongest prediction power of an R-squared of 0.65, whereas for the AD cohort, we have a 0.53. So in conclusion, we have found that each diagnosis group, whether it's MCI or AD, has its own set of reliable regions. Those are the places of high tau deposition with predictable pattern of tau accumulation. We have also found that the network diffusion model can help in 2 ways. One, by providing a model for the pattern of tau deposition, and in another way, by identifying those reliable regions for each of these cohorts. And our results suggest that the structural connectivity of the brain is driving the tau deposition accumulation in Alzheimer's disease. So with that, I'd like to acknowledge our funding source, Eli Lilly and UCSF, and everyone here for your attention. Thank you.
Hello, everyone. My name is Dan Jin from Institute of Automation, Chinese Academy of Sciences. Today, I will introduce our work about generalizable, reproducible, and interpretable imaging biomarkers for Alzheimer's disease. Alzheimer's disease is a kind of neurodegenerative disease, which is different from normal aging process and characterized by memory impairment and cognitive decline. It is the most common cause of dementia. MCI, especially amnesic MCI, is a transitional stage between normal aging and AD. Previous studies have shown that about 3% to 15% of MCI patients will progress to AD every year, about 10 times of the normal elderly. A biomarker is a physiological, biochemical, or anatomic parameter that can be objectively reproducibility measured as an indicator of normal biological process, pathological process, or response to a therapeutic intervention. The identification of underlying biomarkers for accurate clinical diagnosis and quantifying the state of disease is very important. Compared to other methods, structural MRI provides an effective way to characterize anatomical abnormality, making it possible to discover imaging biomarkers of early neurodegeneration. Neuroimaging model development begins with a broad search for traditional predictive models, those with Good diagnostic performance should be further tested for accuracies and generalizability across multiple studies, scanners, and populations. Importantly, most models, about 90%, are still in early stage of development and have yet to be tested beyond an initial development study, and a few models go beyond the second development phase. In recent years, with the rise of deep learning and the growing amount of medical image data, deep learning algorithm has achieved good results in medical image analysis tasks. The research of machine learning methods in medical image have been widely carried out, but there is still a lack of reliable imaging markers in the diagnosis of brain disease. Compared with the traditional machine learning method, the deep learning method greatly simplifies the process of feature selection and feature dimensionality reduction. However, one of the major criticisms is there being black boxes, since there is no satisfactory explanation about how such algorithms arrive at their predictions, which does retard its application in clinical. The existing methods lack the availability of valuable location information or provide in a post-processing manner, which is isolated from the classification performance. In this study, we propose a 3D attention network which achieves the AD classification and gives the most important regions with discriminative ability, improving the interpretability of the model, and systematically evaluates the generalization and neurobiological association of the model. We employed two large structural MRI datasets from our in-house database and ADNI with a total of 1,832 subjects. For the in-house dataset, the images are collected from 6 different scanners of 4 hospitals in China. The 3D AN model mainly consists of 2 modules. The residual module, which relies the shortcut connection of the convolution layer, which avoids the gradient explosion problem. The attention mechanism module was carried out by a convolution layer with a set of filters of 3 cubic kernel sites. The elements of feature map of previous layer are weighted by the corresponding position of attention map to get the attention-aware feature map of the next layer. The value in the attention map reflects the importance of the work source as the corresponding positions, which increases the interpretability of the model. In this slide, we show the performance for AD classification. We evaluate the performance of our method with inter-dataset cross-validation and intra-dataset cross-validation. The mean accuracy for inter-dataset correlation is about 86%. The mean accuracy for intra-dataset cross-validation is about 91%. In addition, we predicted whether patients with MCI in the ADNI dataset would convert to AD within 3 years, with the mean accuracy of 71%. Through the attention mechanism model, we can get the importance of brain regions. For classification, Figure A shows the mean attention score map of regions on the in-house dataset. We can see that the proposed method highlights the brain regions mainly located in the temporal lobe, hippocampus, parahippocampal, cingulate gyrus, amygdala, and prefrontal cortex. The results on 2 sub-datasets have significant correlation. The correlation coefficient is 0.59, which shows that the results have good repeatability. In order to evaluate whether the attention score of brain regions is related to the structural abnormality of brain, we perform a correlation analysis between the significance of the group difference as indicated by a two-sample t-test and the attention score of regions. The result shows that the attention score of regions have significant correlation with the group difference map in both datasets, which indicates that the proposed model captures the features of abnormals region in AD. To evaluate whether the attention score were associated with the patient's cognitive abilities, regional correlation coefficients were calculated to measure the relationship between attention score and the MSE score in the AD and the MSAD group. The attention score of 80% regions were significantly associated with the MSE score for both datasets. These two MSE-associated maps were significantly correlated, which indicates that the result can be replicated across sites. To investigate the clinical relevance of the prediction performance, we perform correlation analysis between the predicted score and the MSE score and the amount of cerebral spinal fluid A-beta, tau, and polygenic risk score of individual subjects. In this case, we use the anomalized predicted score to calculate the correlation. The higher the score, the higher the probability of AD. We can see that the predicted probability of AD have significant correlation with the cognitive impairment, neuropathological changes, and the genetic factor. The result shows that the higher the risk of individual's clinical score, the higher the risk of AD. So in conclusion, the proposed 3D attention network achieved good classification performance without handcrafted feature generation and captured imaging biomarker with an attention mechanism module, increasing the interpretability of the model. The generalizability and reproducibility was cross-validated on independent dataset with an accuracy up to 92%. In addition, significant associations between the classification output and the clinical characteristic of patient provide neurobiological support for the model. Thanks to the lab instructor and the hospital partners. Thank you very much for your attention. If you want to join us, please contact.
Hi, I'm Yashar Zeighami, a postdoctoral fellow from Dr. Alan Evans's lab in McGill University. And today I will present my work on brain disorders and brain disorder categories from a transcriptomic point of view. There has been a lot of new interest in genetic associations with these different brain disorders, and there has been studies that are trying to show how different genetic variants will predispose us to different brain disorders by calculating and quantifying polygenic risk scores that shows different genes can basically predict our future symptoms. Here we are looking at genes from a more functional point of view, and we will look at the gene expressions for genes associated with different brain disorders. In order to do so, we looked at this GeneNet data set, which is the data discovery platform and has different sources of gene disease associations. We only took the gene disease associations that have been reported from one curated source. Then after that, we filtered the disorders that have less than 5 genes and only took the ones with 5 genes and more. After that, we got 200 disorders, which 49 of them were related to central nervous system and brain. Finally, we went through the literature and we added the missing genes from these 49 disorders that weren't included in this GeneNet data platform. These 49 disorders are then categorized based on the literature to 8 different broad categories: psychiatric disorders, neurodegenerative, movement, developmental, and cerebrovascular disorders, substance abuse, brain tumors, and brain-related. And whatever was not in these categories went to the brain-related or other categories. This color code will be used throughout the presentation. In order to look at the gene expression, we use Allen Human Brain Atlas of Gene Expression based on 6 postmortem brains. These subjects didn't have any neurological or psychiatric disorders before passing away. And 2 of them were sampled in both hemispheres, 4 of them only on the left hemisphere, 5 male, and each sample was annotated for 17,000 genes. In total, there was 3,700 samples and from 132 different brain structures. All these transcriptions were normalized across the structures and across the brain. When we were looking at the gene expression, we had 3 different measures that we investigated. The mean expression level, where the question is, do genes associated with brain disorder have higher expression level? The second measure, which will be defined in the next slide, is stability of expression profile over brain structures, and we want to see if these genes do they have higher stability or no. And at the end, we will look at the disease association based on their expression profile and how the expression profile will put the diseases in relation to each other. Gene stability is basically a measure that shows how we can translate the expression profile from one brain to another. In order to define it, we look at the gene expression over all the structures in one brain for one gene, and then we correlate it with that gene's expression in another brain. We repeat that for each of the 6 brains in pairs. So in total, we have 15 unique pairs of brains, and we calculate the correlation for every one of them. And at the end, the average correlation is defined as the stability. This can be done in local and substructure level as well. But as you can see, there is a high correlation between the general stability and the rest of stability measures. So we use this general stability in this work. Mean gene expression. So the question is, do genes associated with brain disorders have higher gene expression level? And we found no significant difference between genes associated with brain diseases versus the general gene population. However, there is a high variability between different disease categories, where neurodegenerative disorders, tumors, and developmental disorders show higher gene expression levels, and psychiatric disorders and cerebrovascular disorders show lower level of expression. However, even within neurodegenerative disorders, we can see FTD and ALS have a high degree of gene expression, where multiple sclerosis, Alzheimer, and Huntington have lower expression levels. For the gene stability, we see that there is a significantly higher stability level for genes that are associated with brain disorders compared to the general gene population. However, again, we see a high degree of variability between disease categories and between disorders themselves. Here, psychiatric disorders, movement disorders, and substance abuse disorders show higher stability, where neurodegenerative disorders, tumors, and developmental disorders show lower stability. And we can see here that the movement disorders and psychiatric disorders are consistently on the top of the sorted disorders. One thing to consider here is that even though there is a variability between them, there is in general a high stability, and most of the diseases have at least 50% of their genes associated with them at a higher level than average gene stability shown here by the red line. So when we compare these 2 groups, the conclusion that we have here is that the higher stability in genes associated with brain disorders might suggest that they have a more specific functional aspect to them in the local level, and when they are not working properly, that causes symptoms to appear. The next thing to consider is gene expression results that come from higher gene expression but low stability need to be interpreted cautiously, because it might not be replicated in different subjects. And at the end, the reason we see like these high gene expression levels might be due to the fact that they have a vital role in the cellular functions, but it might not be specific to certain locations in the brain, and that might cause lower stability in those genes. Next, we moved on to looking at the gene disease association. In order to remove the bias for our approach, we removed the overlapping genes between every pair of disorders that we looked at, and we did a clustering based on the average gene expression for each disease after removing these overlapping genes. Then we correlated that with the other disease that we are comparing, and 1- correlation was used as the distance measure. However, the Euclidean distance has a very similar results that are not shown here. And we used word clustering, hierarchical clustering for our method of clustering. On the left, we can see the average pattern of the diseases in each cluster. We can see that cluster number 2 has more cortical, amygdala, hippocampus, and striatum, and is more consists of psychiatric disorders and movement disorders mainly. However, cluster 3 is mainly tumors and developmental disorders, and they expressed higher in myencephalon and white matter. Cluster 4 is mainly the neurodegenerative disorders. However, when we see, for example, multiple sclerosis in cluster 3, that might suggest that it has more, the genes within multiple sclerosis has more functional and general patterns similar to the tumor or developmental disorders. Next, we looked how reliable is this clustering approach. In order to do so, we looked at the clustering, we repeated the clustering within each of the 6 brains from the Allen Institute. And here we are showing how many times out of 6 each 2 diseases are clustered together. And we can see that there is a high degree of stability for within cluster association between these disorders, which red shows 6 out of 6 times. Then, in order to look at the structures of these clusterings, we use multidimensional scaling, which is an approach that basically preserves the distance used in the clustering while bringing them into an abstract 2-dimensional space. We can see a high degree of separability between clusters, with cluster 2 as outer circle red here, cluster 4 here, cluster 1 here, and cluster 3 here. The other aspect is that there is a nice structure between the traditionally categorized disorders. We can see psychiatric disorders, neurodegenerative disorders, developmental, and tumor related. The other thing to consider is that the MDS1 is capturing the cortical versus subcortical aspects of the separation, whereas MDS2 looks at the more detailed subcortical expression patterns. In summary, we found that there is no significant difference between mean expression levels for disorder genes and the general gene population. However, we see a higher stability level for genes associated with brain disorders. The mean expression and stability vary within and between disorders. Tumors, neurodegenerative, and developmental disorders show higher expression level on average, and psychiatric and movement disorders have higher stability. We should look at the genes with higher stability and focus on them if we want to, generally speaking, study the gene expression patterns, because between subjects we might have a higher variability for other genes. At the end, we showed that the clustering approach that is data-driven has a stable structure to it when it's compared to each single sample clustering. And it has also a similar structure to the disease categories based on system and pathology defined categories. At the end, I want to thank my supervisor, Dr. Evans from McGill University, also this work supervisor, Dr. Havrilich from Allen Institute, and my collaborators, Dr. Bakken and Miller. This work was funded by Healthy Brain for Healthy Lives and Quebec Neuroimaging Network. Thank you.
Hello?
Yes.
Yes. I will begin at 11.
You can start right now.
Okay. Thank you. Okay. I'm right on at here to present my work. My presentation is about discovering propagation pattern of neurodegeneration across the brain network. Alzheimer's is a common neurodegenerative disease with long and dynamic progression. The neuropathology starts from temporal lobe, and then spread to parietal lobe, followed by frontal lobe, and finally end up in the occipital lobe. This neuropathological propagation may be caused for decades. Now, there is no effective treatment for Alzheimer's, so many currently interested are focused on the preclinical studies. Now, recent advances in image technology allow us to measure the neuropathology and the neurodegeneration pattern. To measure this change, we commonly use the 18 biomarker. What's the 18? A refers to the amyloid biomarker, T refers to tau biomarker, and N refers to neurodegeneration. Neurodegeneration commonly includes FDG and structure MRI. All these studies show that amyloid arises at an early stage. When it reaches a certain level, it will induce tau. Tau will kill the neuron, which eventually causes the decline of metabolism. However, the pattern of the pathology burden spreading across the brain region is still unclear. Thus, it's time for us to discover the pattern of neurodegeneration propagation across human brain. As well known, brain network is a powerful tool in the study of Alzheimer's. So we are interested to know how the neurodegeneration propagate across the brain network. As well known, in the network there are a small amount of brain region. We call this region the hub node. They play an important role in the whole brain. So the first hypothesis here is that does Alzheimer's different affect hub node? If this hypothesis is right, we want to know does the Alzheimer's-related neurodegeneration start from hub and propagate across network? And the third hypothesis is that does the propagation pattern follow the connective pathway or spatial vicinity? To test this, our proposed hypothesis, we have collected 139 subjects from the EDNET database. All of these data have T1 MR image, DTI image, and PET image. By applying registration, all these scans are aligned with the T1 MR image. Based on this image, we can get the FDG level, amyloid level, cortex thickness level, and get the structure network. To answer my first hypothesis, we extracted the FDG as a neurodegeneration burden on its brain region and calculate the node centroid on its brain region by plotting the correlation between the node network centrality and the neurodegeneration burden among different groups. As we all know, the node degree and between are two important centrality in network theory. Here, we plot the linear fitting curves of neurodegeneration level with respect to connection degree and betweenness. Well, here, the neurodegeneration level is measured by FDG PET. From the result, we can find that there is a reduced FDG level at nodes that are densely connected. This suggests that this densely node received more neuropathology than the other non-hub nodes. To verify this conclusion, we also plot the linear curves of neuropathology of amyloid. We can also find that there is a high amyloid level at those nodes with a high degree and betweenness. So the Alzheimer's are more easily to affect hub node in the network. Both of these images support our hypothesis that Alzheimer's differently affect hub node. To answer my second and my third hypothesis, we use a linear model to capture the propagation pattern of the longitudinal trend of neurodegeneration between hub region and non-hub region, with the consideration of network distance and the spatial distance. In this linear model, the demographic factor of gender, education, time, diagnosed group, are also included in our linear effect model. The term at the left side is the neurodegeneration change at non-half node, and the second term in the right side is the neurodegeneration change at half node. The third is the network distance between the hub and the hub. The fourth is the spatial distance. This linear model can describe the correlation between the neurodegeneration change from hub to non-hub along network pathway or spatial pathway. If the neurodegeneration spread from hub node along the network pathway, it will have a negative correlation for the third term. Similarly, there will be a negative coefficient for the fourth term if the neurodegeneration propagate along the spatial distance. By fitting this model, we summarize the significance of network distance and spatial distance in our linear model. In this table, with the small p-values, that's the larger influence in the propagation of neurodegeneration event across the brain network, while the second column shows the summary about the significance on network distance, and the third column is the result about the spatial distance. We can see that there are many nodes are significant in the second column, and well in the third, this result is not in the third column. Based on this result, it seems to support the propagation of a neurodegeneration burden largely follow the network pathway instead of spatial vicinity. By doing this work, we conclude the following conclusion. The first is that half nodes are easily to be affected in Alzheimer. The propagation of Alzheimer follows the connective pathway rather than the spatial pathway. So this is our presentation. Thank you.